Thanks for joining us for this symposium. I'm Bill Rapisi, President and CEO of the Lymphatic Education Research Network. LE&RN's mission is to fight lymphatic diseases through education, research, and advocacy. In order to win a fight, you first have to join it. So we ask, please become a supporting member of LE&RN at lymphaticnetwork.org. And we hope you enjoy today's symposium.
Good morning, everyone. Thank you so much for joining us in what we hope will be a very informative hour together about finding a drug to treat lymphedema, and I'm going to add lipedema as well, as you'll see during the presentation. Thank you all for being here. We understand that this is a very robust crowd of over 800 signed up participants, so I'm very honored that you're all here today, and we hope that you have a lot to learn and experience. Before we begin, I just want to acknowledge the corporate sponsors for this event that you see listed on the current slide. I want to also indulge in this disclaimer that we want to be sure that you make any decisions about your healthcare with a healthcare professional, and this symposium is not meant to take the place of healthcare or services from your healthcare providers. So any of your personal health concerns should be addressed by the healthcare providers who care for you. I also want to take the opportunity to announce a parallel event that will occur next month. Many of you know that on an annual basis since the start of the pandemic, I have been hosting here at Stanford a virtual lymphatic summit, and this year is no exception. We will have this summit on February 21st of this year. This will be a 6-hour symposium that will be available both live and in an archived form after the symposium is finished. It is intended for both clinicians and scientists and to those patients within the community that are also interested in learning the leading edge of science in relation to lymphatic health and disease. I believe that the link for registration will be available to you in the chat portion of this Zoom function, so for any of you that would like to sign up for this. So now let's move on to the subject matter at hand. Just to give you an overview of what we'll be doing, I will start with the didactic portion of this presentation. I don't know how many of you will have heard the prior presentations on the background for this drug therapy approach, so I really will go over everything once again, just so everybody's on the same page, but try not to take too long with that. Once I've given you an update on where we stand with the studies, I have the distinct pleasure of introducing you to one of our subjects who will talk to you about her personal experiences within the work and also how she has responded to treatment. And then at the end, we hope to leave enough time to address at least some of the compelling questions that you can put in the question and answer portion of the Zoom function. So I don't think I need to convince this audience that lymphedema is a very compelling problem. It affects at least 5 million Americans, possibly as much as 10 million Americans. And we know that it is a health condition that is debilitating for many, if not most. It is progressive, and historically we have had very appropriate approaches to try to stabilize lymphedema, but we certainly don't have and haven't had treatments that actually will reverse the impact of the progressive nature of this problem. Now, the disease, as all chronic edema does, begins as so-called pitting edema, which means that as the part of the body that's affected accumulates fluid, that fluid is able to be moved physically through the tissues. And so if the examining finger puts pressure on the limb, it leaves an indentation or so-called pit that is the physical evidence that the fluid is present. And we have had for decades appropriate therapies to try to minimize this fluid component and to keep it under control and to hopefully keep it from progressing. And this represents the approach that we call complex decongestive therapy. Sometimes diuretics are used as well in the appropriate context. However, The big problem with lymphedema and the unexplained problem historically is that there is this almost unavoidable tendency to go from pitting to non-pitting, in which case all of the decongestive therapies cease to have an appropriate function. They no longer will minimize limb volume. They will not produce a result. And this represents the stage of lymphedema that most individuals with chronic lymphedema are left with even after they've had appropriate therapy. So CDT will reduce the risk of infection, it will increase mobility, it will increase function, it will minimize the limb volume from its starting point, but we get to a point in the treatment of the disease where about 50% at least of the increase in the volume of the limb is left unaddressed and has no, or historically has had no, therapeutic intervention. When I entered the field in the early 1990s, we had no explanation for why this transformation occurred, nor did we have any appropriate therapeutic approach to try to change the outcome. And that represents where I started my long journey to develop a drug to reverse and potentially, we hope one day, to prevent lymphedema. Now, in order to develop a drug, we have to understand how the disease is behaving, what is the aspect of the disease that will be responsive to therapeutic intervention. And with lymphedema, which is such a complex biological problem, it's hard to know exactly where to begin. And I think this group of scientists evaluating an elephant is a good metaphor for where things stood in the mid-1990s as I began to try to approach this problem. And where you look and how you look intensively will determine what you think the problem is. In our hands, we decided that the big issue with lymphedema was the chronic inflammation that accompanies the disease and, as we have learned, drives the disease forward. In order to try to identify what the relevant features are in lymphedema, I created an animal model in the mouse to simulate what happens in human lymphedema. And while, of course, we want to be very ethical about the use of animals in research, I must say that this model with a minimal impact on animal survival has led us forward in a tremendously meaningful way. What we discovered is that our animal model created changes in the skin of the mouse that very closely simulated what we know happens in human lymphedema, which is to say that the outer layer of the skin, the so-called dermis, increases dramatically in thickness, both because of an increase in the cellular mass of the dermis, as well as in the deposition of proteins and the accumulation of inflammatory cells, all of which drive this thickening, which represents that non-pitting edema that I showed you from the photograph earlier. In order to try to identify what is happening in the skin to drive this process forward, we utilized what was then fairly cutting edge science. It has now been replaced by even more sophisticated methods. But in that day, it drove us forward quite a great deal. And it's called whole genome transcriptional profiling. And basically what we did is to take the skin of mice with lymphedema from the area that had lymphedema and the skin of unaffected mice and run them through a process where we could generate all of the RNA, which are the messages produced by the cells as they respond to their environment and to the existing conditions, to try to determine what in lymphedema is emphasized as a message from the tissues that is driving the disease that would not be present in the normal cells. And basically what that told us, and much to our surprise, because we were looking for signals related to vascular function or to edema itself, but what we found is that almost all of the difference between lymphedema and normal is represented by the inflammatory reactions of the tissue and by the immune response. And this led us to elaborate a model in which chronic inflammation really sits at the heart of the development and the progression of lymphedema, and it has an interplay with a lot of other functions, but the end result is that it drives the fibrosis or scarring of the tissues, as well as the accumulation of fat beneath the skin, which is so characteristic of the non-pitting form of lymphedema. Now, in our work with this transcriptional profiling, one of the signaling messages that we found that was unexpected and that led us down the path that we're talking about today, I'm sorry, let me go back just a moment, is if you can see my arrow, it's this enzyme in the center of the slide, which is called lipoxygenase. Lipoxygenase is an enzyme that liberates and increases the production of inflammatory molecules in the tissues. At the time that we saw this upregulation of lipoxygenase, it was known to be involved substantially, for example, in airway disease like asthma, but had not been described very much in other medical conditions, and certainly not in lymphedema. But we thought this was intriguing, and so we set about, initially in animals and eventually in people, to look at a drug that might impair the ability of this enzyme to be upregulated. This eventually led us to human clinical trials with a repurposed drug that inhibits both lipoxygenase and another inflammatory enzyme that is related to most of the anti-inflammatories that you can buy over the counter in the drugstore, like Advil or Aleve. And basically, the drug that we used is a cousin of Advil called Ketoprofen that inhibits both lipoxygenase and cyclooxygenase. And what we found is that when we exposed patients with chronic lymphedema to ketoprofen, an oral medication, it's a prescription medication that is different from Advil, so don't go running to the drugstore to buy that. Basically, we found that ketoprofen could very dramatically reduce the thickness of the skin as measured by skin calipers. And what we also found is that when we did biopsies of the skin, we actually saw reversal of most of the abnormalities that I showed you earlier on in the mouse skin, which were also present in the human skin pre-treatment. Most of those things reversed toward normal to a very substantial degree on the basis of ketoprofen treatment. Well, now this was very encouraging, but unfortunately, we learned just as we were completing that trial, the FDA decided that the entire family of drugs that includes Advil, Aleve, Motrin, ibuprofen, all of those, and the drug that we were studying, the FDA decided that there was an increased cardiovascular risk with chronic use, which made us a little bit less optimistic about moving forward with ketoprofen as a long-range treatment for lymphedema. So we felt we had to go back to the animal model and determine whether we were interested in cyclooxygenase as the target, or whether we were truly interested in lipoxygenase, or both, because that would determine our path forward. And what we determined is, indeed, it was the lipoxygenase component, the part that is not related to Advil, Motrin, Aleve, and so forth. And this led us to try to understand better how lipoxygenase inhibition would help us in lymphedema. So what we ultimately determined is that it is one of the downstream products of lipoxygenase called Leukotriene B4 or LTB4 that actually is responsible for what happens in lymphedema. And when we specifically blocked the action of the enzyme that creates LTB4, so downstream from 5-lipoxygenase, we were able, in the course of 1 week, to convert what we can call a mouse lymphoscintigram from its dramatically abnormal state, untreated, to one that was virtually normal. We did nothing except give these mice a daily dose of this drug to inhibit that enzyme, and the lymphatic architecture was able to completely restore itself within a period of a week. When we do a procedure in the mice that is very similar to what some of you may have experienced in your evaluations of lymphedema, which is called near-infrared fluorescence lymphography, what we were able to demonstrate and I'll show you this in a moment as this movie loads. This is the lymphedema portion of the tail. This is where the surgical interruption occurred. And in this mouse that received the lipoxygenase, or so the LTB4 restoring drug, you can see restoration of flow in the channels. Perhaps you can see it. It's not easy to demonstrate over the internet, but I can assure you that what we saw is restoration of flow back through the intact channels of the lymphatics across the area of damage simply by having administered that drug. And we gave control subjects just an infusion of saline, salt water, and of course, we did not see any improvement. So it turns out that there are many things that we can try to do with drugs to try to restore lymphatic function, but the only drugs that work are those that reduce the levels of LTB4. So down here, you see what happens when we give ibuprofen, for example. It not only fails to make things better, it actually, in many cases, make things worse. But all of the drugs that directly or indirectly reduce the production of LTB4 in this study were able to significantly reduce tail volume and give us all of the other positive attributes of treatment that we were able to demonstrate. We also showed that in both mice and in human subjects, if you have lymphedema, there is an increased level of circulating LTB4 in the blood, whereas in the normal mice and in the normal healthy human subjects, those LTB4 levels are normal. So further indirect evidence that LTB4 is very appropriately associated with the presence of untreated lymphedema. So again, we were able to eliminate our focus on what I'll call the ibuprofen side of the equation and limit our attention to the production of LTB4 and reducing that production in order to reverse the problems associated with lymphedema. Now, how does LTB4 help us? Well, it turns out, as we learned in this more recent series of animal studies, that LTB4, when present, blocks the activity of various molecular pathways that lead to lymphatic repair. So when we block LTB4 production in an individual who has lymphedema, we can restore lymphatic repair, which gives us optimism that if we can come up with an appropriate drug strategy to treat lymphedema, once we restore lymphatic repair and allow that to happen over a period of time, this may actually prove to be a self-sustaining treatment that will not require chronic administration of the drug, but this remains to be demonstrated over time. The next step in our journey was to test a drug that had many actions in the body, including some inhibition of LTB4 production. And this was, we hoped at that time, a very exciting study. And it unfortunately had to be terminated for sponsorship reasons before we ever achieved the full goal. So we were not able to statistically analyze our results, but I do want to tantalize you with the results of one subject who at the onset of the trial was disabled from single leg lymphedema, as you see here, untreated at the time of entry into the trial. And after 6 months of being randomized to the active drug, and 18 months later, when I received this photograph by email, the leg had restored to fundamentally nearly normal size and was able to be maintained simply with a very light compression stocking and no other treatment. So one success does not equal FDA approval, and we had other successes, but not a completed trial in order to present to the FDA. But we were very encouraged that LTB4 production or antagonism would be useful. So this is a question I get asked week in and week out through my career, and I want to assure you that I'm working as hard as I can to get to the finish line, but these efforts are laborious and very time-consuming. But we are now, as many of you know, in the midst of what we hope will be the definitive human trials that lead to commercialization of a drug for lymphedema. So this study we call the HEAL trial, the Human Lymphedema Acebelestat trial. It is co-sponsored by a granting mechanism through my parent university, Stanford University, as well as the company that owns the rights to the drug, which is called Celtaxis. It is a huge undertaking to establish a phase 2 clinical trial, and you can see all of the things that I have to do to get this trial underway. So it took us about 2 years even to begin the trial. Again, we had the support of the IMA program here at Stanford, as well as Celtaxis. This is a description of the trial that is currently underway. And before I tantalize you with any disappointment, I'm not going to be able to give you any of the results from the HEAL trial itself because we're not able to do any of the objective documentation of the data collected until the entire trial is completed. However, I can say very optimistically that we are close to complete enrollment of that trial, and we anticipate concluding the clinical events by the end of this year, which means that by early in 2026 at the latest, we hope to have some data that we will be able to present to the FDA. Many of you are aware that one of the things that we are doing for the first time in the HEAL trial is to use so-called dermal ultrasound, where we, instead of having to do biopsies, we can actually look at the behavior of the skin, which is the area of the body that is damaged in lymphedema. We can look at the skin by putting an ultrasound probe directly on the surface of the skin and looking at the various layers and making very sophisticated measurements there. So this is an example of what we might see as we do those dermal ultrasounds. Now, I'm going to get to the meat of today's discussion, which is to tell you that because of the safety of ace-bilostat, which is the drug that we are using now, a very highly targeted inhibitor of LTB4 production, because of the safety of that drug and because of the compelling need in these lymphatic diseases, the FDA has given me permission to conduct 3 separate expanded access programs. One of them is for the people that complete the HEAL trial itself, who show evidence of response to the drug. We have been given permission by the FDA to continue treatment in an expanded access program for an additional year. The trial is over, but we continue to monitor the patients for safety and for their response over an additional year of treatment. Number 2, and the one I'm going to talk about the most today, is that they have allowed me to conduct an expanded access program in leg lymphedema. Now, they have specified that this leg lymphedema cannot be cancer-related for reasons of safety, but in the event that an individual has leg lymphedema that meets the other criteria, we have been able to treat these patients for a period of 1 year with the same drug, using essentially the same protocol that we use in the formal trial itself. And that experience, I'm happy to say, is almost completed as well. That is to say, we have enrolled nearly the number of subjects that the FDA will allow us. And the final expanded access program, which is just launching now, is one for lipedema. And we are very optimistic about the potential impact of ace-bilostat in lipedema, because many of the things that we see responding to the drug in lymphedema are some of the shared attributes between lymphedema and lipedema, which makes us believe that lipedema may respond in a very similar fashion. We have the first several subjects for that program lined up, and that's taken us a bit of time to launch that particular program, bureaucratic and organizational issues, but we're going to begin enrollment in that program within the month, and therefore perhaps if I get the opportunity to see you again in several months, we'll be able to have a progress report on what's happening in lipedema. So let me show you what's happening in the expanded access program, because here we are able to look at the data in real time. So I already showed you what happens with the skin calipers when we used Ketoprofen in our very first trial, our published trial. We're seeing responses that are even exceeding those that we saw with Ketoprofen. So you can see that in all but 1 or 2 subjects, the skin thickness by caliper reduces to a dramatic degree. And in general, when we do clinical or any scientific research, We do a statistical analysis to determine how significant the changes are. That's called the p-value. And anything less than 0.05 is considered significant. And you can see that for skin caliper, our significance is way more than 0.05. It's a p-value equal to 0.0003. So highly, highly significant reduction in skin caliper thickness. Also, we are measuring limb volume. And while the Ketoprofen trial was a little disappointing insofar as limb volume did not statistically significantly reduce, even though the skin got much healthier, in this case, we are seeing a significant reduction in limb volume as well. Again, achieving statistical significance, that value of 0.03 is less than the threshold of 0.05, and you can see that over half of the patients had substantial reductions in limb volume over time in those latter two graphs on this slide. And we are also looking at quality of life measures. So it's wonderful if skin thickness goes down with a caliper, or if a patient has some reduction in limb volume, but if that doesn't impact the way she or he might feel, then we haven't really accomplished very much. So we are using an instrument that I have created for this purpose to measure quality of life. And again, you can see the now an improvement in quality of life is represented by an increase in the total score here. And you can see again that our quality of life measures are increasing highly significantly, 0.0001. Finally, one of the things that we are measuring in the EAP that we are not able to measure in the HEAL trial because of the logistics of the equipment involved is so-called LDEX, or bioimpedance spectroscopy. Many of you will be familiar with this technique, but basically it is an ability to detect how much fluid is in the tissues by how readily they allow a very minimal amount of electrical current to pass through the tissues. And basically, when lymphedema is present and untreated, there is an increased amount of fluid, which increases the transmissibility of current because water conducts electricity, and therefore the value becomes increasingly abnormal. And I'm sorry, I went in the wrong direction. So this is what a clinical instrument looks like that is used to measure the bioimpedance spectroscopy. It's called the LDEX. And you can see that this specific individual, drawn from our clinical practice, has had two measurements, both of which are abnormal. There's a slight variation from one to the other, but because these numbers go up and down from day to day, this is not considered a significant change. What we can say is that lymphedema is present on both occasions. What we are seeing in the EAP, the Expanded Access Program for Lymphedema, is that, again, to a significant degree, the bioimpedance spectroscopy is improving with treatment, which indicates that with the drug, the amount of fluid in the tissues, when the patient continues everything that was being done to maintain the lymphedema prior to the initiation of the drug, that number indicates that fluid is being cleared from the tissues on the basis of the drug itself. And again, this does achieve the statistically significant threshold of less than 0.05. Now, the final attribute, the one I'm not going to be able to give you distinct numbers on yet because we're still in the phase of analysis, is to look at these dermal ultrasounds. We do know, very surprisingly and very gratifyingly, that the amount of fat that we see under the skin is reducing in relationship to this drug treatment. We don't have our final analysis on this yet, but I do want you to know that the way we are proposing to look at ultrasound is that we are everybody's talking about AI these days. We've actually created an artificial intelligence platform in which we can feed in the paired images of pre to post treatment and ask the computer to look at all the variables that are present in order to help us understand on a quantitative basis the degree to which improvement has occurred with treatment. And again, when we meet again, perhaps I'll be able to share that data more concretely with you. So here you see a patient after 3 months of treatment. Here's another patient after 6 months of treatment, and you can see the shortening of that red line indicating the degree to which the subcutaneous fat has decreased in relationship to drug. So at this point, I'm going to say that I have concluded my comments, and I'm going to have the great pleasure to introduce you to one of the subjects in the Expanded Access Program who has graciously agreed to come and talk to you today, to introduce herself to you, and to tell you about her lymphedema story and about her experience within the EAP. So Carrie, I'm going to leave it to you, and I will turn off my video so you have center stage.
Thank you. And Dr. Rockson, do you notice my special Zoom background? Hope is the most important medicine, which I'm going to share why I included this quote in a moment. But hi, everyone. I am so honored to be here. My name is Carrie Smith. I am one of the participants in Dr. Rockson's Expanded Access Program. And I'm here today to tell you a little bit about my experience and give you a peek into what it's been like. But first, I wanted to start by sharing my story and my background. This is such a full circle moment for me. I was diagnosed with lymphedema, primary lymphedema, by Dr. Rockson when I was just 14. So over 20 years ago, I woke up one day and my mom said, "Did you break your ankle?" And I said, "What are you talking about?" She said, "Your leg is swollen." And like so many people with lymphedema, I searched for answers and couldn't find them. And finally was connected with Dr. Rockson, got the diagnosis, and began to resume, how do I fit this into my life? How do I treat this? How do I take care of this? And for the first few years of having lymphedema, I would say it was relatively manageable. I could pretty much hide it. I just started wearing pants. I would wear, compression stockings, of course, and do the manual lymph drainage. And it was pretty much taken care of. But over the years, it really progressed and really started to affect my life. I went into college and noticed that my leg would get more swollen. It would be a lot harder to recover. My skin started getting thicker. I was dealing with more serious issues like cellulitis infections. And it became something that was so clearly going to be a huge part of my life and not just something that I could sweep under the rug. And, then I came into my 20s and I really started to take active control over my lymphedema and started doing everything that I could to try to minimize the swelling and to really to try to chase any sort of solution. I met with all the doctors. I explored some Western medicine. I would try anything. At one point, I even tried to see a healer. I tried chiropractic work, acupuncture, lasers, all sorts of things, anything to try to relieve the pain and discomfort that I was in. I even knew that, I wanted to become a mom, but I wanted to avoid becoming pregnant because I was fearful of passing lymphedema on to kids. And I wanted to do everything that I could to take care of my lymphedema before having children. So I did that. I had surgeries on surgeries on surgeries. We're really fortunate to be able to access those because I know so many lymphedema patients aren't. But those surgeries are grueling and they're tough. And I did experience some relief from those surgeries, and and then I was able to have children. I have two beautiful kids, a boy and a girl. They're 2 and they're 5. And I was just sort of humming through life, just like accepting that lymphedema was going to be part of my life, that I would have it kind of as under control as I could possibly have it. But there was always this underlying feeling of this can't be it. there has to be more. And randomly one day, my husband, who is amazing, he was looking at some scientific papers and he stumbled across something that Dr. Rockson had published. And he learned about this study, and he learned about this program. And I put everything I could into being part of it. And I was so lucky to get in. And here I am today. I'm sorry, it's so emotional. I think I cry every time I see Dr. Rockson. He knows this. I'm 10 months in here, and I can say that this has been just the most amazing experience, that I feel so much hope for my lymphedema. Hope is the most important medicine. Dr. Rockson, I don't know if you remember, you said this to me the first day that I came to you for this study, for this program. And I had so many questions for you. I'm like, what is this? What's going on? Tell me about this. This is a drug to treat lymphedema? Like, this is crazy. I had so many questions, and I'm sure you all do too. And he said the most important thing is that you have to have hope. And I think that is what makes this experience so amazing. This team is so amazing. They care so much about lymphedema. And my drug experience has been pretty great. I have been on the drug for about 10 months now. And, you know, I have really had no side effects, no issues. I can live my life. You know, I know that it's different for everyone, but I really have had a great experience, and my lymphedema is better than it's really ever been. There was pretty immediate changes within the first few weeks, and those changes have continued throughout this year. My skin is noticeably less thick. My leg bounces back so much quicker. It's never getting that swollen anymore. I feel more comfortable taking breaks from my compression stockings. I just feel more comfortable in my skin. And mentally and psychologically, I just feel happier and more relaxed. I feel the sense of hope that I never had before, because lymphedema is no longer that thing that is in the back of my mind holding me back. And so, you know, I am just eternally grateful for this experience. And I just want to say to all of you who are either in this community or you're a patient, that hope really is there. That I know that it's hard because you want that immediate relief, and that you want that instant cure, and that you know, want access to this medicine yesterday. But, you know, hang on to hope, because it's coming. And this drug has made me feel so empowered to take care of myself and my leg and as best as I can. And it's given me a new lease on life and given me a boost of just positivity that I needed, because this drug, this condition is just such a drag on a day-to-day basis. It is something that, you know, I've had for over 20 years, and for the first time, I just feel lighter, and I feel just this sense of hope. So hang on to that hope. I'm so grateful, and I'm happy to answer any more questions that you might have. But that is what I wanted to share today, and thank you for listening.
Carrie, thank you so much. And I know everybody on this Zoom symposium is going to be eternally grateful for you for sharing your story. For those of you that will have specific questions for Carrie about the experience within the Expanded Access Program or anything else, please direct them into the Q&A, and we will have time at the end to answer questions from either one of us. I do want to emphasize, Carrie, that I'm very well known for making people cry, so you're not alone, but I'm glad everything is turning out well.
Yes. When you are going to see Dr. Rockson, don't wear mascara, pack tissues. It's going to be a whole thing.
Absolutely. I'm going to go ahead and share my slides again because I do want to end by you giving one additional testimonial from within the participants. This was sent to us by email, and I'm not going to name this individual, but I do want to read to you what she wrote and show you a couple of pictures. So this subject said, I wanted to send a quick email with some progress. I feel like my leg always looks more swollen after traveling to my visits. She lives out of state and comes by plane to the visits for our program. But I wanted to provide some pictures of some progress while being on this medicine. Over the past 16 years, I have lost so much mobility in my leg, foot, and knee. After having my daughter 2 years ago, I was having issues falling. I was incredibly unstable on my leg and having balance issues. It was difficult and painful to squat down to play with my kids, usually resulting in me falling over. I've noticed a life-changing difference since being in this drug study. My mobility in my right leg has made a 360. I can squat down, as you see here. I can stand on my toes. I can sit on my knees with my legs bent under me. She can also do a crow pose in yoga. I can move my toes and actually bend them. They have even popped during one of my MLD sessions. My toes haven't popped since I was a kid. These are all things that were incredibly challenging prior to the drug study. It's easy to forget small changes as they happen quickly, and it's easy to forget what it was like prior, and we take these easy things for granted until we can't do them anymore. I've tried for over a decade diligently to get back what I used to be able to do, and this drug study has significantly propelled my advancement towards my mobility goals. Some other successes since starting the drug trial. I believe I mentioned at last appointment, we're being able to fully run both a 5K and 10K in 2024. I had a goal of being able to run a full 5K since 2016, and I was never able to meet that goal until after starting the drug trial. In the past, my calf muscle would constrict and hurt so badly, I couldn't run for that long of a distance. And that's with wearing compression. Also, another success, I joined an adult cheer team. That's the patient on the left-hand side up in the air. It's not something I had intended on doing, but it came back in the fall of 2024. I did competitive cheer in grade school and high school and had to quit when I was 17. I didn't cheer my senior year of high school as I lost a significant part of my mobility. So just another thing to show the drastic change in my mobility. These pictures are on a public website for cheer, so again, no issues with sharing. Anyways, hopefully this can make it into feedback on the drug's progress. The change in my mobility has made a significant difference in my life. Thank you. So I think that is what I have to share. And again, I firmly believe in this statement that anything is possible, and we are so gratified that we have come as far as we have with this program. I hope that this is giving you optimism, because we hope to convey information of this type to the FDA in the near future so that we can move on within the commercialization process in order to one day have this drug available to anybody on this call that needs it and wants it, as close as the corner drugstore. So with that, I'm going to invite Carrie back, and we're going to move to the Q&A session, and I'll try to get through as many of these questions as I can. Please know that there are many of you here, and we're certainly not going to have time for everything. Let's see. So somebody wanted to know, are these results for primary or secondary lymphedema or both? So the HEAL trial is primary and secondary lymphedema, but it is the upper extremity. It does not have to be cancer related, but as you can imagine, almost all of our subjects are breast cancer survivors. For the expanded access program that Carrie participates in, This is, again, primary and secondary. The only caveat is these are not cancer survivors. We're not allowed to have cancer survivors in this. There was a question about any known cardiac side effects. No, that is the big distinction between this drug and Ketoprofen, which belongs to that family of NSAIDs that has been earmarked by the FDA as predisposing to cardiac problems. Ace-Bilostat belongs to a completely different category of medications, and to our knowledge, there is no effect on cardiac function whatsoever. A question, are we still looking for participants for these drugs? For the lower extremity expanded access program, we are nearly replete, but I would not discourage you from contacting us. And if there's still ability to enroll you and you meet the criteria, we're happy to think about that. In fact, I do have a slide, I think, that I forgot to share, which I will do now, that has contact information. So I'm going to leave that up so people can look at that while we are talking, in case you are interested in any of that material. So for the HEAL upper limb trial, again, that is upper extremity unilateral lymphedema. Again, we're near completion there, but if you have a compelling interest, please let us know, because we can see if it can still happen. The middle portion there is the program that Carrie and the other individual I quoted have participated in, and that is non-cancer related primary or secondary lymphedema of the leg, not the arm. And lymphedema protocol, which is just beginning and has the most access, is the bottom one, and Osama is the coordinator for that particular program. So I have a question from an attendee in the UK who's asking, at what level of lymphatics are we seeing repair, superficial, deeper, or even nodes? I don't think we have the sophistication yet to say that with certainty. I think based upon the near-infrared fluorescent lymphography in the mice, we can say that there is some deep lymphatic repair because we are seeing flow through the deep lymphatic channels. But whether this is a repair mechanism that occurs throughout the lymphatic tree, whether it is driven by the areas in which there is obstructive physiology, there's still a lot we have to learn about that. Somebody wants to know, is CDT still the most effective treatment until now? Yes, it is the baseline core treatment that everybody mandates, whether or not you have surgery, whether or not you are going to be taking a drug. At least in our experience to date, we are using Ace-Bilostat as another layer of therapy on top of what the patient is already doing, which means we ask them to continue all of their self-care. In order to be in one of our programs, you have to have completed a course of CDT and be at least 8 weeks out from that in the maintenance phase. And we are building on that with additional benefit. Now, one day, will we be able to supplant some of that physical treatment? Very possibly. Number two question, do we need longer than a year to see the ultimate benefit from this drug? Very likely. And hopefully some of our expanded access work will begin to help us understand how long treatment benefit continues to accrue as patients take the drug continuously. And certainly we hope that one day this drug category will be able to be used in a preventive capacity in individuals who are at high risk for developing lymphedema. And there, if we were to preemptively treat to stimulate lymphatic repair before any lymphedema appears, then hopefully we would never get to the stage of actually needing the CDT. Somebody wants to know if this correlates in any way with the Ozempic type drugs. No, this is completely different. And I think the Ozempic part of things has to do with the very poorly understood but easily recognizable interplay between adipose biology and lymphatic biology, so that when the adipose responses are not normal, we know that the lymphatic responses can be affected, and therefore, if Ozempic helps to reverse some of that adipose pathology, we think that indirectly we see benefit in the lymphatic defect as well. So along those same lines, somebody else asked about my thoughts about Ozempic. I think that the case reports and the subjective reports are encouraging. We still do not have the kind of carefully controlled observations that we're trying to do in a phase 2 trial. And at the end of the day, in the practice of medicine, we consider evidence-based medicine to be reliant upon that kind of very rigorous evaluation with statistical comparisons to non-treatment or control treatments. And so I think we should continue to think about drugs like Ozempic, but eventually we do need to formally test it as well. Manual therapy is, again, as I said before, the mainstay of our therapeutic approach. Somebody asked about colchicine. We have not tested colchicine, but I want to clarify, colchicine does not directly block LTB4. It does have some impact indirectly at a very subtle level on 5-LO, the parent enzyme. So it's a little more like Ketoprofen than like acebelostat. And I think that the differences between acebelostat and Ketoprofen that we've seen to date make us think that this is a much, much more potent therapeutic than what we were seeing initially with Ketoprofen. Is the drug effective with lymphedema? Well, that's going to be the question we are going to address in the expanded access program, which has not yet begun. By inference, because we know that acebelostat improves lymphatic function, which we know is impaired in lymphedema, and because it shrinks the layer of adipose beneath the skin, which we know is the locus of problem in lymphedema, we're very optimistic that it will be effective, but we don't know that yet. Somebody asked whether lymphedema changes to lymphedema. No, it doesn't change to lymphedema, but it can become secondarily complicated by lymphedema. So it is a form of secondary lymphedema that is seen in combination with lipedema when this does occur. It does not occur in all lipedema patients, but it certainly has the capacity to do so. Somebody asked about side effects or adverse reactions from acebelostat. That's a very compelling question. I think Carrie helped you understand that side effect was not a problem in her experience. And actually, in our array of patients that we've been treating in all of our programs, side effects have basically not been an issue. When they occur, they are extremely transient at the onset of treatment, perhaps a day or two of very non-specific symptoms of the type that we see really across the spectrum of any prescription drug, some gastrointestinal things, perhaps headache, or maybe some visual disturbance, or very, very subtle minor issues that tend to self-resolve within a day. We have not had to remove anybody from the program or even reduce dosage because of symptomatic side effects. Now, I do want to mention that acebelostat, like all drugs, have to be detoxified in the liver, in the body and eliminated. And in the case of acebelostat, this is accomplished by the liver, and we are aware of the degradation pathway that is utilized by acebelostat in the liver. We are very cautious to not have drugs that will interact with acebelostat in that same pathway. So if those drugs are being administered, we will need those to be discontinued, or somebody is not eligible to take the drug. We do watch liver function very closely, and we try to adjust dosing such that we know that the liver is happy with this. So it's not very different from any of you that are taking a statin for high cholesterol, for example. It's a very similar process of watching the liver to be certain that the liver is able to cope with the dosage that is being given. And in almost all cases, this is the case. Let's see. So somebody wants to clarify that expanded trial for leg edema does not enroll patients whose lymphedema resulted from cancer treatment. That is correct. Somebody is asking about the recommended ketoprofen dose for congenital lymphedema in children. I cannot give that dose because I consider that to be an experimental therapy. I'm not saying that we don't continue to use it on rare occasions, but because the full evaluation of ketoprofen did not go on to a more phase 3 trial, and because of the warnings of the FDA regarding ketoprofen, we have to proceed really cautiously. And I would say that would be a question for you to undertake with the assistance of your healthcare provider. Can this medication be useful for lipodystrophy? That is a difficult question because in the case of lipodystrophy and lipedema, there is a lot of a naming convention that makes it hard for us to understand what we're looking at. If we're looking at lipodystrophy as a form of lipedema, or what others would call lipedema, then yes, it could potentially be useful. But there are other forms of lipodystrophy that have a completely different mechanism of development, or what we call pathogenesis, and there I really cannot answer the question. Okay. I'm just looking to see if there are any questions for Carrie, because we're getting to the end of our hour and I want to really be able to utilize her.
Yeah, I've been
strategy.
answering a bunch of questions in the chat.
Oh, good.
Yeah, so thanks everyone. And I think you've answered a lot about, side effects. I think the only thing that somebody asked about, like, kind of getting started in the community, are there any books? I was blown away by like how many support groups, how many activists, how many nonprofits are so actively out there online. And I don't know, Dr. Rockson, if you have a book recommendation for people. I don't think I've read some like nutrition books and things like that, but I would say go on Instagram, go on Facebook. There's so many support groups. There's even lymphedema influencers. That's a thing,
Yes.
which is really cool. We can talk about like fashion, and we could talk about like being a mom. Like there's all kinds of things for everyone. So definitely get out there. You're gonna find the support and the connections. And that will also really help you on your journey.
I will add that I think LE&RN itself is a tremendous resource for information in all aspects of lymphatic disease, including lymphedema. So certainly utilize heavily the LE&RN webpage and its resources for every piece of information that you need. One last plug that I will make, I think this is relevant to the comments here, and maybe we'll end with this, is to make sure that everybody knows that at the behest of Congress, the National Institutes of Health has created a national commission on lymphatic disease, which is heavily focused on lymphedema along with the other lymphatic diseases. I have the honor of serving as the co-chair of that commission. We are we're about halfway done with our 2-year commission of assembling information and resources and input from stakeholders. And at the end of our 2-year commission, we will be making a report to the NIH, which will then be transmitted based upon its approval to Congress about what should be done to improve the lot of individuals in the United States with lymphatic diseases. And I can tell everybody today that we have been heavily focused on lymphatic education and resources for the lymphatic disease community, as well as education of healthcare providers, so that the healthcare community is prepared to deal with the needs of lymphedema subjects and patients, which we all know has been woefully deficient until the very present day. So I hope that you will look with a similar degree of hope at what can come out of the National Commission, because I think we're looking at the next half decade as being a time of tremendous growth for the diagnosis and treatment of lymphatic disease, including lymphedema. So at this point, I want to thank Carrie. Thank you once again. You've added immeasurably to this event. I want to reemphasize the Virtual Lymphatic Summit on February 21st and encourage anybody who has an appetite to learn about all of this to learn even more. We'll go into much more depth on all topics. You can use my talk as a coffee break time because it's going to be very much what you've already heard today, but there'll be plenty of other information on a variety of topics. And I want to thank everyone for your attendance, and please hang on to that very, very important element of hope. So thank you very much. Bye-bye.